So I'm in Nicholas Bernier's lab doing my 4th year research project. He mostly studies stress physiology in fishes. So my project is going to be looking at how temperature affects the early ontogeny of zebrafish and how that affects their ability to develop a hypothalamus pituitary entorhinal axis. So just a bit of background knowledge. The way that an animal perceives a stress is through its HPI or HPA axis. In fish, it's HPI. So as they develop, they're first provided with a maternal deposit of cortisol, and then as that sort of depletes with their yolk sac, they have to begin their own de novo synthesis of cortisol through the HPI axis. So what I'm looking at is how temperature can affect that development. So a lot of research has been done in adult fish on sort of acclimation temperature or acute temperature changes, on how that affects basal cortisol and a fish's ability to respond to stress, but no study's been done during early ontogeny. So that's what I'll be looking at. So my basic hypothesis, I have my first experiment, which is going to be looking at just basal cortisol. So when I raise larvae at 3 different temperatures, which will be 25, 28.5, which is their optimal raising temperature, and 32, I'm just going to be looking at basal cortisol. How is there any changes affected there? My second experiment will be looking at after 5 days of raising at those 3 temperatures, how they can respond to a stressor at the 5-day mark at that exact temperature. So that sort of thing has been shown in adult fish where they've been acclimated to a temperature and then a stressor occurs at that temperature. And typically what happens is in higher temperatures, fish are able to mount a stress response faster, and then they're also able to clear the amount of cortisol quicker as well. And then sort of the opposite with the lower temperatures, it takes longer to respond to the stress, and it also takes longer to sort of clear that amount of cortisol. So we would expect to see the same sort of thing at that early stage 5-day development, so long as the suboptimal rearing temperature hasn't negatively impacted their ability to respond to a stressor. And then the third experiment that I'll be doing is I'll be raising them at the suboptimal temperatures till 5 days, and then putting them back to the optimal temperature, and then raising them back to 10 days, and seeing if they're plastic enough to be able to sort of, if the HPI axis is plastic enough to sort of bring them back to being able to respond to a stressor in a normal way. So in order to study early development, you need to spawn the fish. So zebrafish are a really great choice for doing these types of experiments because they're easy to take care of. They spawn several days a week if you treat them right, and when they do spawn, they have a very quick development cycle. So you can kind of look at the whole early ontogeny within the first 5 to 7 days. We set up tanks of fish and we put in like an egg barrier, and then you also include marbles and some pretty fake plants to kind of, so it's called environment enhancement. And then you put the fish in there, you set them up overnight. We have them in a water bath so that they're still kept at the right temperature that they should be. And then when the morning comes along and the lights turn on, that's actually what triggers spawning. So then the fish can release their eggs. And then at the end, when I come in, I collect the fish, I put them back in the system, and then I can remove the egg barrier and we'll see nice eggs sitting at the bottom, and we can collect all those and put them right into the incubator for the proper temperatures. So after I've collected the embryos and they've raised them to the certain days that I want to collect them at, you can collect them on dry ice, and then you bring them up to our lab at Nick's lab in the science complex, and then you kind of just freeze them into little tubes. So you need 30 larvae for 1 sample of cortisol. And then you can do a cortisol radioimmunoassay, which involves sort of extracting the cortisol from the larvae and then purifying it through a column. And then I use a radioimmunoassay to quantify the amount of cortisol. So this project can be pretty important for a couple different reasons. One of them simply being how labs choose to rear larvae for stress experiments. So for example, if we find that even the slightest change in temperature really is detrimental to their development of the HPI axis, and people are planning to raise larvae and then later do experiments with them, it'll be an important finding. So a lot of really interesting research has been done on mammals recently on multi-generational effects. So when something detrimental happens to the HPI axis of a mother, and then the offspring are then born, and not only that generation, but the generation following, you can see subsequent molecular effects on the HPI axis. So it's really important to see how these temperature changes, especially with climate change, are going to be impacting fish's ability to respond to stress.